Two ways you can do this. One is, this is pretty obvious and super lame why we're doing this. The second is we're getting out of the lab, stretching our legs a little bit. Okay, can anyone, and it works because the board is empty, can anyone draw a spectral curve for us? Sarah did it last time. She did a very good job, I agree. Would anyone else like to take a shot at drawing a spectral curve, just roughly, for this? Anybody?
Do you have different colors, or is it all
You
one?
know, if you're gonna be so
There's some
demanding, I don't
colors.
know if I would
Oh,
have
there's
called on
some.
you.
Oh, wow.
Just for like red,
I like to do it right, you
blue,
know?
green, red, near infrared, just the general stuff here. Okay, let's see what Lauren can come up with.
Magic, that's what I'm coming
All
up
right.
with. So this is my soil color, because there's no brown.
Okay.
Okay. Isn't it like that? It's really high for soil.
You can just do veg if you want.
Oh.
Yeah, you can just.
I thought all three.
Go for it. If you want to do all three, that's just fine.
There's a little bumpy do. Oh, it's a little too low. Ooh.
I think we have enough of these.
No, we'll just do that, and then this is your water. It's pretty low. It's like that.
I like it. Very, very
Okay.
good. Yeah. Very,
Interpretive.
very, very good.
Okay.
Okay. Thank you, Lauren, for doing that. Now that Lauren has done that, I have a very simple question. Lauren's got this vegetation curve on the board here. We've got 3 different types of plants right here. They're all green, more or less. But they all look like Lauren's spectral signature.
No.
People seem very confident about this. Anyone disagree with the group here?
I think it looks similar.
Parts of it probably will, but overall, probably different. Well, which ones would look different?
Of the green ones?
Yeah.
Oh.
Yeah, don't even worry about it. Just focus on this stuff right now.
Probably the pine. River pine.
The hemlock, this one, the
Yeah,
conifer.
yeah.
Why would that look different?
It's not maybe capturing as big of an area of green, or reflecting as big of an area of green.
Yeah, no, I like it. Could you tell me like the subtleties of how the curve would look different?
Probably not. But
Probably not. I'd probably
maybe a little bit lower of a green reflectance than the other broadleaf plants.
Perhaps, yeah, perhaps. So point being is, yeah, they'd probably be a little bit different. We might not know how they play out, and that part is not important with respect to this. It's that they could be a little bit different. And keep in mind, right, we've got, this is a single leaf, and this has like, I don't know how many M&Ms, it has many, many, many leaves on there as well. If we cut cross sections and we put them under a microscope like we had for those general leaves, they would look different. Our leaf area is different. Probably going to manifest itself to some differences in our spectral signature when we scale that all the way up. That's sort of the general thing I wanted to sort of talk about. This leaf, this plant in particular, this leaf has a pretty thick cuticle, right? So it's much more rigid than some of these others. But that leaf architecture is a really big deal because it means the cells are, they're kind of the same, but they're going to be different in a needle-leaf versus a broadleaf plant. Okay, so we can do that. All right, next question. So what is phenology? What is, can you give us a definition of what is phenology? Victoria, you look like you might know the answer.
I probably don't. I would take a guess studying plants.
Study of plants? Okay. That's not correct, but I love the guess. And it very much applies in the study of plants. But let's think even more broad than plants in particular. What?
Is phenology just the study of like death things?
Close. It's really just the timing of biological events.
Okay.
Right? So phenology, when it comes to plants, is how things sort of green up, get really green, and then they're gonna start to yellow and drop their leaves in the fall. Phenology for butterflies could be, for monarch butterflies, is the time of year that they migrate. Same thing with birds. But yeah, I did mention the phenology of death. That's sort of unique to like mountain pine beetle kind of stuff. Okay, so that said, we've got two hollies right here. No, I don't want to say that yet. We've got a holly, a hemlock. This is actually a species of oak. Would these look different at different times of the year?
Yes.
Yes. Why would they look different at different times of the year?
Well, some trees lose their leaves.
Trees lose their leaves. Absolutely. So what you're saying, when we've got no leaves on here, no photosynthesis, it's going to look very different. Yeah. So when we think about how can we relate phenology to remote sensing to sort of aid us in detection of different types of plants, perhaps? Anyone?
If you're looking at like different growing seasons, if you're not looking at something that would just like it'll look everything will look much more dead essentially
Much
compared
more
to like
dead.
the growing seasons.
Yeah, it sounds, I mean, it's comical, I think, and it's so obvious. No, that's a really big deal, right? When it comes to Landsat, we talk about trade-offs between spatial resolution and temporal resolution. Yeah, we might be losing something in sort of our spatial resolution, but we're gaining all that power in temporal resolution. So we can look at an area, we can look at CDNP in the high winter, as long as it's cloud-free, snow-free, and say, oh, here's an area that's loaded with conifer trees, right? Because it doesn't look nearly as different. So we can, if we're building some kind of a statistical model, or even if we're just doing a supervised classification, which we're working on right now, we can do it high growing season, we can do it high leaf off season. It's a very, very common thing that a lot of my students use in different models. Even if you're trying to, like, let's say you're trying to look at changes throughout the fall in the broadleaf community. Well, part of what makes the fall so great is not everything turns the same time, not everything is the exact same color, they're different. And you can actually tease that out if you've got enough shots going through it. Okay, so last one here is, these are the same plants. One looks pretty good, one doesn't look so hot. How would the spectral signature be different between these two plants? Say this is our standard one, right? How would it change when you go from the green to the yellow? Anybody remember?
A lower infrared curve at the end, so
Yep, lower infrared curve. Is that the only spot where we would see differences?
no, wouldn't the red also go up?
Absolutely. And why does the red go up?
Because there's less red absorbance.
Because there's less chlorophyll actually. Absolutely. And we know, right? You know, if someone said, wait, would these look different in the blue, the green, and the red? Absolutely. The blue, the green, and the red is the visible portion. And they all might look a little different to all of us, but generally, I think hopefully everyone can tell that those are very different leaves. And that's what we're seeing go on is the mesophyll, especially the spongy mesophyll, right? This is still very much a leaf. I mean, I broke it there, but other than that, like it basically it's still rigid. Like there's still stuff going on in there, but the reason that coloring is looking different is because the spongy mesophyll is starting to break down. It's really no different than in the fall with our oak trees as they start to senesce. Even though it's a time senescence, it's basically the same thing, right? We're losing that ability to do photosynthesis. So when people do studies of phenology, there are lots of studies of phenology that go on not just to figure out what species are there, but is phenology changing over time, right? Mike, what would be an example of a phenology study? Like why do we think it might be changing over time, or why do we care? And it doesn't have to be about plants; it could be about stuff.
Oh, well, I was just thinking maybe shifting climates over time and how that changes the timing of sprouting of leaves or the dying off of the leaves.
Absolutely.
Yes.
Absolutely, yeah. So when we talk about this timing of different events, we can look at data and say, when does this area, when does Northeast Ohio typically green up, right? And we can actually measure that based on the slope of a curve. When does it green up? How long is it green for? And then when does it start to decrease? And we can actually do that with, we can do some kind of like greenness, we'll say. I know it sounds silly, we are going to talk about that more. And over here is day of year. So here is 1, that's Jan 1. And then, 150, day 150 is like, what, mid-May? And so basically it's gonna look something like, that's basically our growth curve. We can generate that from satellites. If we look at that every year, we can start to say, is it changing? And as we all know, living in a place like this, we get tons of variability, right, from year to year. It's pretty hard to remember.
Exhibit A.
Yeah, like how are things really changing? But when you start to put together that long-term, decadal measurements of this, you can start to see those trends, and we can say, oh, you know what? The growing season, it's actually getting a little bit bigger, a little bit longer in places like this. Maybe here, that's not a terrible thing. Other places, it probably is, right? And the cascading effects of that, we could spend a decade discussing all the research that's going on with respect to that. So we might get, like this year, we're probably gonna get, unless it really changes soon, we're gonna get an earlier green up. That doesn't necessarily mean that, oh my gosh, everything's changed because of this one year, but we are seeing increased variability. And maybe that's in some areas. We can't say anything definitive about those changes, but we could at least say there's way more variability than there used to be, which is also of a concern. But in all these kinds of studies, right, everything that we're dependent on is basically the mesophyll, the cells in these plants, that is helping us to sort of see those changes over time. Okay, we got enough time to get back to the classroom. Any questions about the Super Duper Phase 2 field trip or anything we talked about? Thanks everyone. Let's make our